Cyrus Tan from the Quay Morris Lab in MSK. I would like to share about my work on using protein language models on target prediction on nucleic acid binding proteins. So nucleic acid binding proteins includes both DNA and RNA binding proteins, and they recognize specific sequences and structures. We could use microarrays to measure the binding preferences of different proteins. For RNA binding proteins, we have RNA compete, where we incubate proteins with a pool of RNAs, pull them down, hybridize them to a microarray. And for each protein, we can assign a vector, so-called RNA 7-mer Z-scores, which describes the affinity between each protein and all RNA 7-mers. For DNA binding proteins, we could do a similar thing using protein binding microarrays. However, out of all proteins, only a tiny fraction of them have any measurements. Therefore, we need a method to assign binding preferences to the unmeasured ones. Protein language models could generate protein representations based on sequences. These representations have been proven useful to predict protein functions. However, the use in predicting binding preferences of proteins are unknown. Therefore, we assayed the representations generated by a bunch of different protein language models coupled with different predictors across a wide array of DNA and RNA binding proteins. What we found is that protein language model outperformed the baseline across different predictors and data sets. You may have heard of these structure-based models such as Rosetta Fold NA and AlphaFold 3, which could fold both protein and RNA or DNA sequence simultaneously. So we compared protein language models against Rosetta Fold 2 NA. And what we found is that although Rosetta Fold 2 NA could predict structures, they're actually not great at predicting whether a protein or DNA or an RNA bind together. Out of all protein language models, we find that ESM2 works the best. Therefore, we decided to fine-tune ESM2. We incorporated protein and nucleic acid sequences as its input and fine-tuned ESM2. We find that compared to the pre-trained model, fine-tuning ESM2 actually helps with the performance significantly. So if you're interested in my work, come see my poster at A321 today and tomorrow. Thank you.